In this video, we will discuss about glycosylation that will trigger the cell death, and we will discuss two different cases. First, in the tumor cells, and another one is the immune cells. And from our previous videos, we understand that this apoptosis can be triggered in the cell either through mainly two different pathways. First one is the extrinsic pathway, another one is the intrinsic pathway. And this FASL receptors and trial receptors helps in triggering the immune apoptosis within the cells. So however, in the tumor cells, because of the mutations, this glycosylation machinery get disrupted, that will lead to the aberrant glycosylation on the cell surface, and that will cause different effect. Some of them might help in the attenuation of the apoptotic pathway, and some of them might trigger apoptosis pathway. So in the FASL receptor, there is a aberrant glycosylation of alpha-2,6-sialic acid, and that will prevent the apoptosis by affecting the downstream signaling pathways such as this extrinsic pathway through this caspase 8 and caspase 3 will not be activated, because FASL receptors will not be able to interact with the extrinsic factor because of the aberrant glycosylation in the form of this alpha-2,6-sialic acid. And however, this aberrant glycosylation in the aberrant glycosylation trial has a different function. One of the function is that it will with the help of extrinsic galactin 3, it will trigger apoptosis by forming this FADD disc complex, and that will trigger this activation of caspase 3. It will trigger extrinsic apoptosis. And this galactin 1 intercellular galactin 1 will also trigger apoptosis by disrupting this AK13 phosphorylation. Thereby the survival of the tumor cells will get disrupted. And this galactin 3 has intercellular galactin 3 has two different roles. First one, when it is present in the free form or it interacts with the ceramide, it will trigger intrinsic apoptosis, and also it will disrupt this AKT PK13 complex formation, and therefore it will disrupt the survival potential of the tumor cells, and on the other hand, it will also trigger intrinsic apoptosis. However, galactin 13 has also been found to attenuate extrinsic apoptosis by dysregulating this FAS downstream signaling pathways by interacting with the ligand that are present on the intercellular part of the FAS. So this galactin 3 has found to be has multiple roles. One is to trigger apoptosis with the help of triggering intrinsic apoptosis, and also by disrupting this survival pathway of P13K and AKT. At the same time, it will also attenuate the extrinsic apoptosis that can be triggered by FAS receptors by dysregulating the FAS ligand within the cells or within the cytoplasm of the cells. So this is how the glycosylation work in the tumor cells. Now let us understand how this glycosylation works in the immune cells. In the immune cells, these FASL receptors are usually related to the apoptotic signaling pathways. So this aberrant or this glycosylation in the FASL receptors will prevent the interaction with this TNF, and also this alpha-2,6-sialic acid prevent the interactions with this galactin 9, and thereby the downstream apoptosis will not be triggered because of the non-efficient interaction or non-interaction of these FASL receptors and the TNF and galactin 3. And thus also this alpha-2,6-sialic acid prevent this downstream complex formation, that is FADD or FADD disc complex formation that will trigger apoptosis, this extrinsic apoptosis. However, some of the glycans that can trigger apoptosis is this when the immune T cells are in resting state, these extracellular galactins can trigger the extrinsic apoptosis through this caspase A extrinsic pathway, and also this glycophospholipid by interacting with the glycophospholipid by interacting with these FASL receptors can trigger intrinsic apoptosis pathway through this MOAP signaling pathways, and via mitochondria can trigger intrinsic apoptosis. So in the case of this resting T cells, this apoptosis can be triggered via galactin or the glycophospholipid, and this can usually happen to prevent the autoimmune response by overpopulation or overcrowded population of these immune cells. So I hope you got an idea what this glycosylation is related to cell death, both in the case of tumor cells and the immune cells. If you need to understand more about this glycosylation and the cell death or apoptotic pathway, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.